To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.
Hello my dear students, welcome to today's lesson. So we are discussing inheritance. In the previous chapters we discussed what the inherited characters are. Common inherited characters, rare inherited characters, and you are familiar with some of the characters that are developed during the lifetime that do not inherit or they do not get inherited by the next generation. Thereafter we discussed in detail how Mendel, father of genetics, carried out his experiments using garden pea plant or the Pisum sativum plant and why he chose that plant. Thereafter we saw how we can use symbols to explain the occurrence of the first and second generations with Mendel's experimental results. And you learn how to explain that occurrence. Then we discussed the Punnett square. After discussing that, we used probability to explain the ratio obtained by Gregor Mendel. That is, for the first generation, it's all the same dominant character. For example, if it is tall and short plants, all are tall plants in the first generation. That is for a monohybrid cross of pure breed short and tall plants. But in the second generation, you get tall plants and short plants in the ratio of 3:1. Always dominant is to recessive character 3:1. Whereas you saw the factors. There are two factors that determine a character. So they are the factors. If it is tall, capital T is to capital T simple t and then simple t. That ratio was always 1:2:1. So that is what we have discussed so far. And we were able to explain that using probability, using beads of different colors or tossing two coins simultaneously. So I'm sure you all have a good understanding about that concept. So now I'm going to move on to gene concept, or where we explain Gregor Mendel's observation using genetics. So I told you all, when Gregor Mendel he did the experiments, he did not have knowledge about DNA or what genes are, what chromosomes are, because that time the technology was not developed enough. But as father of genetics, he was able to explain everything very clearly. All his concepts are very correct and they were explained very well. Only thing, he did not know about the gene concept. So now we will try to understand using the concept of genetics. So basic concepts of genetics. Gene concepts about inheritance. Now you have heard this, no students? When you have a characteristic, let's say your complexion, color of your complexion, color of your skin, or the nature of your hair, or maybe your structure, the body structure. Then they say, okay, you have inherited from this, from your father, or they say these are the genes of the mother. Sometimes they say these are the genes of the grandmother. So like that, they use the term. We use the term genes in our day-to-day life also. But already, you're familiar with the structure of cells. So you know what the nucleus is. In a eukaryotic cell, there is an organized nucleus. And the nucleus is surrounded by the nuclear envelope. And inside the nucleus, there is the chromatin fiber or chromatin body, which is made up of chromosomes. And these chromosomes are again made up of DNA molecules. You are familiar with that also when we discuss biomolecules. We have learned that DNA stores genetic information and transmits it from one generation to the other. So that information that is stored in chromosomes, and the chromosomes are made up of polynucleotides, that is DNA molecules. So these polynucleic acid chains part of those are responsible for different characteristics. And that is what we will be discussing as genes. So that is just the introduction. Now we will go into detail. So when we say gene concepts about inheritance, now you remember Mendel said that the features of an organism is determined by a special particular factor. So what is this special particular factor? That is what we will be discussing as genes. Now we already used letters. Now we said there are two factors. Let's say capital T or capital T, then simple T. Those are the factors. Capital T is the factor responsible for the dominant tall character. Simple T is the factor responsible for the recessive short character. So these factors are what is known as genes. Is that clear, student? So the identified particular factor is named as genes later. So that is very important. We call this as genes. The identified particular factor is named as genes. So T and T, capital T and capital T, two genes responsible for the dominant tall character. Simple T is the gene responsible for the recessive short character. So now the factor will be referred to as genes because we are learning the concepts of genetics. In gene concept, as a standard, the dominant feature. Now we have already used this. The dominant feature is denoted by a capital letter and the recessive by a simple letter. So that is how we did. Now we used capital T for the dominant tall character. So dominant feature is denoted by the capital letter, whereas simple T is for the recessive character short. Is that clear to you all? So we have already used these letters in Mendel monohybrid cross. So if we look at some of the features, the examples, so you can remember dominant and recessive characters. So if you remember, we used tall as capital T. Short, we did not use S, instead we used simple T. Then if you remember color of flower, purple and white. Purple is the dominant color and white is the recessive color. So here purple flower. Purple flower, so capital P. And white flower, we used simple P. We even looked at round seeds and wrinkle seeds. Then the nature of the pod, color of the pod. So like that, we looked at so many features. Basically 7 features, 7 characteristics that were observed by Gregor Mendel. So for all those, you can use this symbol. So their dominant feature is denoted by a capital letter, these, and the recessive character is denoted by a simple letter. But the capital and simple letter are the first letters of the dominant character. So if it is again round and wrinkle seeds, round seed, we use capital R. Whereas wrinkle seed, you all know we use simple R. Always the letter is the first letter of the dominant character. So to show dominant, it's capital letter, recessive, simple letter. Since we have used these already, I am sure you are familiar with this. So do not confuse. Do not use 2 different letters. Always the same letter, capital and simple. So those represent the genes. So now you know capital T means gene responsible for tall character, which is dominant. Capital P gene responsible for the dominant character purple color flower. Then capital R, the dominant character responsible for the round seed. Then on this side, short is a recessive character, so simple T. White color flowers, recessive character, simple P. And wrinkle seed, recessive character, so simple R. So those are to show the genes that are responsible for those characters. So always there is a pair of genes, two factors. So two genes that are responsible for one feature. Height of the plant is determined by two genes. Color of flower is determined by two genes. I'm sure you all can understand that. So with that, students, we'll move on to the next slide. So here we have homozygous and heterozygous conditions. Now I'll explain this with the same example that we used before. We had capital T combination. Dominant characters, both the genes were of the same type. That gives rise to a tall plant. At the same time, if you take the combination where we had capital T, simple T. Now both the genes are not similar. They are different. Capital T represents tall character, simple T represents short character. But the plant becomes tall, the dominant character. So the genes are different. So if both the genes responsible for a character are the same, then we call it homozygous. But the two genes that are responsible for a particular feature are different, then we call them as heterozygous genes or heterozygote. When we write the combination of gametes, we call it heterozygote, homozygote, or we can refer to the genes as homozygous genes and heterozygous genes. So this is something that you all have to know, students. So if we define homozygous genes, so here we can say if two genes are similar for a given feature of an organism, then it is known as homozygous. Homozygous genes. What are the examples? Purebred, tall plant. How do we write it? Capital T, capital T. Then we had short plants, that is also purebred. Short plant, we had simple T, simple T. So here both represent tall character, similar genes. Here both represent short character, similar genes. So this is a homozygous tall plant, whereas the other one homozygous short plant. Then, if we define heterozygous, so here we can say if two genes are not similar for a given feature of an organism, then it is known as heterozygous. So you can remember this easily, students. You know the meaning of homo, similar. Hetero, different. Zygous, zygote. Zygote is what? Formed from the combination of two gametes. So, male gamete and female gamete combine to form the zygote. So, heterozygous or homozygous means the combination of genes. Homozygous, similar genes. Heterozygous, different genes. So, can you think of an example here? If we take this particular character, the same tall plant. Example, heterozygous, tall plant. So the genes have to be different. Capital T, simple T. But still, the dominant character will be expressed in the generation. So you have the heterozygous tall plant, capital T and simple T. So we had the pure breed, tall and short plants. They were homozygous. The heterozygous tall plant, it has a mixture of tall and short characters, but the tall character will be expressed. Is that clear? So like this, similar to this, can you all think of the other characters where you have the homozygous and heterozygous instances? So if we say if we have the feature homozygous and heterozygous instances, we can write a few examples. So here, feature, we can have homozygous, then heterozygous. So out of these, we have already written for tall and short. So if you take round seed, what is the homozygous instance? Since this is the dominant character, capital R, capital R. Do we have a heterozygous instance? Yes, that is capital R, simple R. Then if you take the wrinkled seed, that is homozygous because it's a recessive character. Recessive characters are always expressed in the homozygous state. So we have simple R, simple R. There is no heterozygous instance. Is that okay? Same goes for, let's say, the color of the flower. Color of flower, purple flower. Purple flower, white flower. So purple flower, capital P, capital P. It has a heterozygous condition. Capital P, simple P. But still it is the purple flower. Why? It's the dominant character. But white flower, only the simple P situation. Now if we look at some more characters, a few more characters. A few more examples. So here again, we had the round seed, wrinkled seed, purple flower, white flower, then color of the pod. green pod, yellow pod. So, green pod, that is the dominant one. So, we have capital G, and heterozygous instance, capital G, simple G. But still the character that is expressed is green color pod. Whereas we have the yellow color pod. Yellow pod. So like already all know, yellow pod is the recessive character. So we have the simple G, that is the recessive nature, homozygous recessive character, that is when the color of pod becomes yellow. This also does not have a heterozygous thing. Then we can write down one more character. We have used the tall and short character, round and wrinkled seed, then purple flower, white flower, green pod, yellow pod, and you can remember the color of seed. They are also yellow and green, but the yellow seed is dominant, green seed is recessive. So you have to remember that, student. They will normally give you the features, but you have to remember. Green pod is dominant, whereas the yellow color, I will use a different color to write. Yellow seed, that is dominant. And green seed is recessive. So for this, of course, we will have to use capital Y for the homozygous dominant character. Then heterozygous dominant character, capital Y, simple Y. Whereas green seeds, it will be simple Y. There is no heterozygous condition. So from all these examples, you all can see for the heterozygous situation, wrinkled recessive character, there is no combination. Again for white color flower, recessive character, there is no combination. Yellow color pod, recessive character, there is no heterozygous combination. And green color seed for recessive character, there is no heterozygous combination. Same for the tall and short plants. Short plant, there is no heterozygous combination. So that is something you have to remember. So here, if we write that as a note, dominant character can be expressed by homozygous or heterozygous genes. But the recessive character is expressed only by homozygous genes. So these are things you will have to take into consideration, students. Something you need to understand and you have to remember. Always, because the dominant character, that is what dominates over the recessive character. So because of that, it can be either the homozygous condition or the heterozygous condition. So that can be expressed. But if the recessive character has to be expressed, it has to be the homozygous condition. So there is no heterozygous condition for a recessive character to be expressed. That is something you have to remember. So if we quickly go back and see what we discussed. So we try to understand what homozygous and heterozygous are. If two genes are similar for a given feature of an organism, then it is known as homozygous. For example, pure breed tall plant, capital T, capital T. Short plant, that is also pure breed, simple T, simple T. Now also you can see. Then, the heterozygous condition, if two genes are not similar for a given feature of an organism, then it is known as heterozygous. Example, heterozygous tall plant. So, we don't have a heterozygous short plant. There is no heterozygous recessive character. You have to remember that. Then here, I gave you all a table where we looked at a few examples. So, feature homozygous, heterozygous condition, round seed, capital R, capital R, capital R, simple R for heterozygous. Then wrinkle seed, only simple R, homozygous condition. Purple flower, capital P, homozygote, capital P, simple P, heterozygote. White flower, simple P, homozygote because it is the recessive character. Then green color pod is the dominant character, capital G, homozygote, capital G, simple G, heterozygote. And yellow pods, simple G, only the homozygote is expressed because it's the recessive character. Then yellow color seeds, capital Y, that is the dominant character. Capital Y, simple Y, that is the heterozygote instance because it is the dominant character. Heterozygote also gives rise to the yellow color seeds. And green color seeds, simple Y because it is the recessive character. It is only expressed in the form of homozygote or homozygous condition. Homozygous genes. Genes both are similar in character. Is that clear to you all? So there, I am sure you all can understand this part also. Dominant character can be expressed by homozygous or heterozygous genes. But the recessive character is expressed only by homozygous genes. I am sure you all can understand that. In the next slide also, I will be again recalling what we discussed here and thereafter we will move on to gene expression. So here, example for homozygous and heterozygous genes. What we have already discussed, like I told you all, I'll be going through it again. So gene for round seed. It can be capital R, or it can be capital R, simple R. Because round seed is the dominant character, it can be the homozygous or heterozygous combination. But gene for wrinkle seed is only simple R. Is that clear, students? So here, homozygous situations, if we look at different examples, now like I discussed before, we can say purple flower, capital P. Then white flower, simple P. You can even take the color of pod. That is again green color is dominant. So we'll say green, capital G. Yellow, simple G. Same goes for round seed. Capital R, round seed. Wrinkle seed, simple R. That is also homozygous situation. So here, these two are homozygous and this one is heterozygous. same as these. Capital R, simple r, simple r, same similar genes. Both of them represent the same character, capital R and capital R, round character. Simple r and simple r, wrinkle character. So those are homozygous. Whereas capital R, simple r, capital R represents the round character, simple r represents the wrinkle character, but the expressed character will be the dominant character, round seed. So that is a heterozygous instance or situation. So here we are looking at more examples, homozygous situations. Purple flower, capital P, capital P. White flower, simple P, simple P. Color of pod, green color is dominant, so capital G, capital G. Yellow color, pod is recessive, simple G, simple G. If you take the heterozygous situations, now for tall plant, capital T, simple T. Then round seed, capital R, simple R. Green color pod, capital G, simple G. Then we have the purple flower, capital P, simple P. These are all examples of heterozygous conditions or heterozygous situations. And you can see in all these, green color pot, tall plant, capital T, simple T, round seed, capital R, simple R, green color pod, capital G, simple G, purple flower, capital P, simple P. All these are dominant characters. Now, dominant characters are expressed when there is a heterozygous situation. Is that clear, student? So then we will move on to the next slide. Gene expression. Now, gene expression. So far we looked at the gene combinations. What genes are and how they represent a factor. We did not go into what genes are made up of or the components that store the genetic information. We haven't gone into that. But you all know gene is the factor that was discussed by Gregor Mendel that is responsible for the expression of a particular character. And if the genes are similar, we call it homozygous. If the genes are not similar, we call it heterozygous genes. So now we will see how gene is expressed. So combination of genes is what we call as gene expression. Combination of genes responsible for a particular feature. The combination of a gene pair for a particular character is known as gene expression of that organism. So this is something very important you all have to remember, students. When we say gene expression, the combination of a gene pair of a particular character is known as gene expression of that organism. So for any character, there can be 3 different gene expressions. What are those expressions? You can remember. Now for tall or short character, that is the height of plant. We had tall and short. What were the gene expressions? We had capital T, then we had capital T, simple T, and we had simple T, simple T. The 3 types of gene expressions. A pair of genes for a particular character. Combination. You can have 3 combinations. Capital T can combine with capital T can combine with simple T can combine with simple T. Now you can understand. From the first generation to the second generation, we got all these 3 combinations. So the combination of genes responsible for a particular character. That is what we call as gene expression. So if you take the shape of seed, again the same set of characters, students. Shape of seed. What were the gene expressions? We had the round and wrinkled seeds. So round, capital R, capital R. Then we had capital R, simple R. And we had simple R, simple R. The three possible combinations for a character, that is the shape of the seed. Round seed, wrinkled seed. So this is a homozygous dominant gene expression. This is a heterozygous, again dominant gene expression. Or we can say heterozygous round seeds. This is a homozygous wrinkled seed. So if we separate them, we can write them in that manner. Now for example, if we denote this is homozygous dominant character. So capital R, simple R, this is a heterozygous dominant character. And here we have homozygous recessive character. So if we highlight all three, this is a homozygous dominant character. Homozygote round seeds. Then we have the heterozygous round character or heterozygous dominant character. And here we have the homozygous recessive character. Homozygous recessive character. So can you all understand that? And you can always remember dominant character can either exist as a homozygous situation or a heterozygous situation. Whereas the recessive character only exists as the homozygous situation. And the recessive character is expressed only when it is a homozygous situation. So that is the gene expression. Combination of a pair of genes responsible for a particular character of an organism. So that is what we call as gene expression. So you have to remember that as well. The combination of a gene pair for a particular character is known as gene expression of that organism. So we have looked at so many examples. I am sure you can write similar examples using the other characters. Now Mendel carried out experiments where he observed 7 different distinct characteristics. So for all those characteristics, you can write the homozygous dominant situation, heterozygous dominant situation, and the homozygous recessive situation. We have written that before also. We have been writing it again and again. So I am sure you are familiar with all these characters. So with that, students, I will move on to the next slide. Phenotype and genotype. I have explained this term before also. Now again going back to the Mendel's experiment. You can remember he used the parent generation, pure breed tall plants, pure breed short plants. So when they were allowed to cross-pollinate and produce the next generation, that is the F1 generation, all the plants were tall plants. So that tall character that was expressed is what we see That is known as the phenotype of a particular organism. The character that is observed or expressed by a pair of genes. That is what we call as phenotype. And the gene pair that is responsible for that character is what we call as genotype. So you can remember the round and wrinkle seeds. If it is capital R, that gives rise to the round character. Round character is the phenotype that is observed, whereas capital R combination is the genotype. Wrinkle seed is the phenotype and the simple R combination is the genotype. So those are two terms that you have to remember. Phenotype and genotype. So here the feature, so you can see the first one, the feature that externally appears is known as phenotype. Phenotype. And the gene composition, the next one, the gene composition to determine that feature is known as genotype. So the character, the feature that externally appears is the phenotype. So tall plant, short plant, round seed, wrinkle seed, purple flower, white flower. Those are all phenotypes. Whereas the gene combination or gene composition to determine that feature is known as the genotype. RR, capital R, simple R, or capital G, simple G, capital T, simple T, those are all genotypes. So these are all terms and concepts related to genetics. Now as examples. Examples, the phenotype of round seed heterozygotes. What is the phenotype? Now round seed is dominant character. What is a heterozygote? Capital R, simple R. But the dominant character will be expressed. So therefore, it will be the round seed. That will be the phenotype. So here it's going to be round seed. Or you can write it as round. Phenotype, round. That is the character or feature that is observed, externally seen. Round seed. And the genotype is going to be capital R, simple R. Why? It is the heterozygote. So, a combination of round and wrinkle character genes. That is the heterozygote situation. Then the second one. The phenotype of round seed homozygotes. Again, phenotype of round seed means this is also round. That is going to be the phenotype. And the genotype, since it is homozygote, it will be capital R, capital R. The third one, the phenotype of wrinkle seed homozygotes. The phenotype of wrinkle seed homozygote. So, if we say wrinkle seed homozygote, it has to be wrinkled. And you all can remember, this is the recessive characteristic. And then the genotype will be simple R, simple R. Is that okay? So, phenotype of wrinkle seed homozygote is wrinkled. The genotype is simple R, simple R. Is that clear to you all, students? So, like this, again, we can write for different characters. So, here, if we use a table, If we make a table like this for a few characteristics, you all can also try it on your own, students. So here we can have the feature, or we'll say example. Then we can have the phenotype. And we can have the genotype. Phenotype and genotype. So, example we can say homozygote tall plants. Homozygote tall plant. So, what will be the phenotype? It will be tall. And genotype is going to be capital T, capital T. Then if we have homozygote short plant. So, can you all guess? Phenotype, short. And what is the genotype? Simple T, simple T. So, by now, you all should remember what is the dominant character and what is the recessive character. Tall plants are dominant, short plants are recessive. Then if we have, let's say, the third type. Okay, we'll have the heterozygote tall plants. Heterozygote tall plant, what is the phenotype? That is also going to be tall. And the genotype, capital T, simple T. So, we will do another example. Maybe the green and yellow color of the pod. Green color pod is dominant, yellow color pod is recessive. So, if we say homozygote green pod, that is going to be the dominant character. So, green color. Then what is the genotype? Capital G, capital G. If you take the homozygote yellow pod, what is the phenotype? Yellow color pod. I will write only the color here, or yellow pod you can say. And the genotype will be simple G, simple G. And finally, if we take the heterozygote condition. Zygote green pod. What will be the phenotype? Green pod. And the genotype, capital G, simple G. Is that clear now? So again, you can understand the same concept. There you can see homozygote. It can be either the tall character or the short character. Homozygote tall plant, homozygote short plant, but heterozygote only the tall plant because that is the dominant character. So that is how we write the phenotype and genotype. Phenotype of tall can be capital T, homozygote. Capital T, simple T, heterozygote. Then if you take the homozygote green pod, green is the dominant character, so phenotype dominant, green, and genotype capital G, because it is homozygote. Then homozygote yellow pod is the phenotype, the feature that is observed, and simple G is the gene combination. And heterozygote green pod, again green is the dominant character, so that is the phenotype that is expressed, but the genotype, gene combination is going to be capital G, simple G. So these are some common characters that I have been using again and again, students. you all can do the same thing with the rest of the characters as well. Even the axial and terminal flowers, inflated and constricted seeds, for all those you can try this. Always use the first letter of the dominant character. If it is dominant, capital letter. If it is recessive, simple letter. So far, under gene concept, you all know genes are the factors that are responsible for the expression or the formation of a particular characteristic. And under that, we looked at what heterozygote and homozygote are, or heterozygous and homozygous conditions. If it is homozygous, both the genes are similar for a particular characteristics. If it is heterozygous, the genes are not similar for a particular characteristic. That is homozygous and heterozygous situations, or what we can call as homozygote and heterozygote situations. Then we looked at phenotype and genotype. Phenotype, the feature that is observed externally or expressed externally. Tall character, purple flower, axial flower, anything. Inflated seeds, inflated pods. So those are all features that are observed or expressed externally. So those are known as the phenotype. And the gene pair that is responsible for all these characters are what we call as genotype. So you all have to understand all these four terms clearly, students. Then if you are given any type of example, you will be able to identify all these easily. I'm sure you all can do that. So with that, we'll move on to the next slide. So next, we will try to understand the nature of genetic material and genes. So when we say genetic material, again I started off with that introduction. You know the structure of a cell. An eukaryotic cell has an organized nucleus. And inside the nucleus, what do we have? The chromatin fiber. That is the chromosomes. And you know, in man, all the cells have 23 pairs of chromosomes. That is a fixed value because we have discussed this with mitosis and meiosis, haploid and diploid cells. So when we have pair of chromosomes, these chromosomes are made up of DNA. That is deoxyribonucleic acid. And this DNA molecule is the one that contains all this genetic information. So when we say genetic material, that is DNA. So from your previous knowledge, students, you all know that in all the organisms, that is plants, animals and man, there is DNA. That is the molecule that stores the genetic information. But in bacteria, you all know there can be either DNA or RNA. And their RNA also can store genetic information. But that is only in bacteria. But all the other organisms, it is DNA, deoxyribonucleic acid, that stores the genetic information. So that is what is known as the genetic material. So the chromosomes are made up of DNA. And in the chromosomes, there are genes. So we will try to understand that step by step. So nature of genetic material and genes. Scientists have identified that the deoxyribonucleic acid, that is DNA, present in chromosomes act as the genetic material that transmits features of organisms from generation to generation. So this is something you already know. So this is a concept that you already know. So you are familiar with deoxyribonucleic acid, that is DNA, and that is the genetic material that transmits features of organisms from one generation to another generation. And this DNA molecule, you are familiar with that. It is made up of nucleotides, polynucleotide chains. So you are familiar to that. But the structure of DNA, it is a double helical structure of DNA. That is something new to you all. Double helical structure. We can call it double helix or double helical structure of DNA was discovered by two scientists, Watson and Crick. So the double helical structure of DNA was first discovered by two scientists named Watson and Crick in 1953. So this is also very important. You have to know that. You need to understand this. So deoxyribonucleic acid, that is DNA, that is the component that stores the genetic material. And it has a double helical structure, and that was discovered by Watson and Crick. Now if you remember, we discussed this when we discuss biomolecules. What are the elements that make up DNA? Elements in DNA, what are they? Carbon, hydrogen, oxygen, nitrogen and phosphorus. And there are three components. What are those components? Phosphate group, then we have the pentose sugar. What is this pentose sugar? That is the deoxyribose sugar. And what is the other one? The third component. That is nitrogenous base. Nitrogenous base. And here also there are 4 types of nitrogenous bases. Can you all remember the names? Adenine, that is denoted as A. Guanine, that is denoted as G. Then there is thymine, T, and cytosine, C. So these are the 4 nitrogenous bases. So what happens? We have the phosphate group. This is how it combines. No, you all can remember that. There is a phosphate group that combines with a pentose sugar. And thereafter we have the nitrogenous base. So what are these? This is the phosphate group. Then we have the pentose sugar. And then nitrogenous base. All these 3 together, what is it known as? It is a nucleotide. Nucleotide. Then what happens? These nucleotides combine together and form polynucleotide chains. So they form polynucleotide chain or strand. We can say strand. So if it is the deoxyribose sugar, then we call the nucleotide as deoxyribonucleotide. So when there is a combination, the polydeoxyribonucleotide gives rise to the deoxyribonucleic acid. So what happens there? All these nucleotides combine and form the polynucleotide. Then that forms the nucleic acid. Nucleic acid. Now, if you take the DNA, DNA, that is deoxyribonucleic acid, is made up of 2 strands. So that is something you have to remember, students. This is made up of 2 strands. 2 strands. That is polynucleotide chains. 2 chains. And these chains are complementary to each other. And you will see in one, I will show you the diagram in the next slide. You will see in one strand, if the pentose sugar is this way, in the second strand, the pentose sugar is inverted. This is the oxygen at the top. Here you can see this is the oxygen. Here the oxygen is facing upwards. And in the next strand, the oxygen will be facing downwards. And always the nitrogen bases combine with each other. That is how the 2 strands are combined together. And once they combine, you can imagine this similar to a spiral staircase. Spiral staircase with 2 hand rims, where you hold your hand and climb down or up. So that is similar to the DNA molecule is similar to that. That is why we call it a helix structure. So you have 2 strands combined together, and like a pair of ribbons being rotated, these strands also form a helix structure. That is the structure discovered by Watson and Crick. So these are all what you have learned before. You are familiar with the components. Now DNA molecule elements are carbon, hydrogen, oxygen, nitrogen and phosphorus. And that combines to form the phosphate group. So here it is the phosphate group. Then pentose sugar, that is deoxyribose sugar, and nitrogenous base. There are 4 types of nitrogenous bases. And out of these 4 types, there is a certain way in which the bases combine. Always adenine is supposed to join with thymine, and cytosine will join with guanine. Or thymine will join with adenine, whereas guanine will join with cytosine. That is how you get the complementary base pairs. So there we have a DNA molecule. And part of the DNA molecule with a certain arrangement of these base pairs is what we call as a gene. And that particular arrangement of base pairs, a certain order, is responsible for a certain feature of an organism. So part of a DNA molecule is what is known as a gene. Is that clear to you all, students? So here, like I said, if we have a DNA molecule like that, these are the 2 polynucleotide strands, and they combine by forming bonds between the nitrogenous bases. So an arrangement, a particular arrangement of nitrogenous bases responsible for a particular feature of an organism is known as a gene. So you have to understand that phosphate group, pentose sugar, and a nitrogenous base combine and form the nucleotide. Nucleotides combine and form the polynucleotide strands. Two such strands combine together where bonds are formed between nitrogen bases. Here, this is just a diagrammatic representation, students. I will explain the structure in the next slide again. So, in this, you will have a certain order. Now, you all know adenine combines with thymine. Cytosine combines with guanine. So, like that, there will be a certain pattern. Let's say adenine, 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 cytosine, cytosine. Then on this side, there will be thymine, 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 guanine, guanine. That particular segment or part of the DNA molecule that is responsible for a particular feature of an organism is what we call as a gene. Is that clear to you all? So, in the nucleus, there is the chromosome, pair of chromosomes. Chromosomes are made up of DNA molecules, and DNA molecules have this particular structure, and segments of the DNA molecule responsible for a particular feature is what we call as a gene. Is that clear? So, with that, I will move on to the next slide. So, here students, this is for extra knowledge, but if you can understand this, you will get a better idea about what genes are. So, this is something that I have already explained also. Here you can see a DNA molecule is a clockwise twined double helical structure of two strands. So, like I told you all, two polynucleotide strands, they twine, they become like a helical structure. A clockwise twined double helical structure of two strands. So, you know what clockwise is? Clockwise turn, and also it's a helix structure that run anti-parallel to each other. Anti-parallel. That is what I told you all. If you look at the diagram, here look at the pentose sugar group. The pink color one, the oxygen faces up in this strand. Whereas in the second strand, the oxygen is facing downwards. That is the anti-parallel nature. The shape of molecule or the structure of the molecules are opposite to each other. They are parallel but opposite in nature. That is why we call it as anti-parallel to each other. The two strands are joined by adenine, thymine, cytosine and guanine, nitrogenous base pairs. So, here also again you can see adenine with thymine, thymine with adenine, thymine with adenine again. Then if you look at the bottom part, guanine with cytosine, cytosine with guanine. Another thing you can notice, here they have shown two lines. That is because there are two hydrogen bonds between adenine and thymine, whereas between guanine and cytosine or cytosine and guanine, there are three hydrogen bonds. Is that clear? So, two bonds between adenine and thymine, three bonds between guanine and cytosine. These are all properties of a DNA molecule. So, anti-parallel strands. And then there are hydrogen bonds and there are nitrogen base pairs. A combination, a particular arrangement of nitrogen base pairs are responsible for a particular character of an organism. That is what is known as a gene. So, when after the two strands combine like that, now here you can see the clockwise helical structure. Now, here one strand and this is one strand, this is another strand. So, they are intertwined together into a clockwise helical structure. We call it double helical structure because there are two strands intertwined together. Is that clear to you all? So, then these combinations in DNA are denoted as ATCG. A for adenine, T for thymine, C for cytosine and G for guanine. Is that clear to you all? So here you can see two strands with sugar and phosphate groups and base pairs. So here C and G base pairs. And in between you will have the sugar and phosphate groups in the strand. Is that clear now? So that is the structure of a DNA molecule. And in relation to that, you should understand what chromosomes are. Chromosomes are made up of DNA, deoxyribonucleic acid. And part of this DNA molecule with a certain arrangement of base pairs responsible for a certain feature of an organism is a gene. So you are familiar with genes and how they are expressed. So this is where that genetical information is stored. So DNA is the genetic material. And the double helical structure of DNA was discovered by Watson and Crick. So those are all information that you all should remember, students. So with that, I will move on to the next slide. According to the sequence of base pairs in DNA strand, different genetic information are stored. So this is what I told you all. So according to the sequence of base pairs, important. Sequence of base pairs. Like I said, the arrangement. Now in the previous slide, you all saw this. Now here you can see A, T, T, G, C. That is a sequence of bases. On this side, T, A, C, G, complementing that. So this whole set, whole arrangement is a sequence of base pairs. AT combined, TA combined, TA combined, GC combined, CG combined. A sequence of base pair that is responsible for a feature of an organism. So the features of organisms are determined by the sequence of base pairs. Again, the same concept. So the feature of an organism is determined by the sequence of a base pair. Accordingly, a gene is a specific base sequence in DNA molecule responsible for a particular character. So is that clear now? What I explained. So a gene is a specific base sequence in a DNA molecule responsible for a particular character. In other way, gene is a specific segment of DNA for a particular character. You can explain it in both ways. Both mean the same thing. Gene is a specific segment of DNA responsible for a particular character. So everything depends on the arrangement of nitrogenous bases and the combination of them, adenine with thymine, cytosine with guanine. So that is the base pair sequence. Very important term. Base pair sequence responsible for a particular character is known as the gene, or a segment of a DNA responsible for a particular character. Now can you all understand that? Again, I am telling you all the same thing. I am repeating it so that you understand it clearly. Inside the nucleus, you have the chromosomes. Chromosomes are nucleic acids, DNA, that is deoxyribonucleic acid. It's a DNA molecule. DNA molecule is a double helical structure in which there are base pairs, and a certain sequence of base pairs responsible for a particular character is what we call as genes. So genes are an arrangement of base pairs. These genes, there are many genes present in one DNA molecule. One nucleic acid, deoxyribonucleic acid, has lot of genes arranged in that. Such DNA molecules are what we call as chromosomes. And these chromosomes exist in pairs. And in a human cell, a cell of a human, a man, all the cells in our body has 23 pairs of chromosomes. So you can relate it now. Is that clear to you all? So if I continue, the genes that determine large number of features in an organism and transmit them from generation to the next are present in chromosomes. So here you hear that term also. They are present in chromosome. So each gene has a particular location in a chromosome. That is also important. Each gene has a particular location in a chromosome. So let's say the characters observed by Mendel. Tall and short character will be present in a certain position. So if you take two chromosomes, now let's say if it is a pair of chromosomes, let's say it is like this, a pair of chromosomes. Now let's say this is character 1 located in one chromosome. The same character will be located in the other chromosome also. We'll just assume tall character, the character that is the height of the plant, tall or short. So these two are the two factors, the two genes. Then let's say round seed and wrinkle seed, the shape of the seed might be located somewhere here. Like that, different genes have different arrangements. This is just an example, students. I am not giving you all the exact chromosome pattern of a garden pea plant. I am just explaining how that will be. So, if these two genes are responsible, when we say these two genes, a segment of the two DNA molecules, the chromosomes, they are always paired. That is a gene responsible for character 1. Then there will be another set of genes in both the chromosomes responsible for another character. So that is how the genes are situated. Each gene has a particular location in a chromosome. Is that clear? And when we have a pair of chromosomes, this component that connects both the chromosomes is known as a centromere. We will be discussing that again also. This is known as a centromere. So, those are the components that you need to understand. Is that clear to you? So, I'm sure you can understand up to here. So, if we quickly go back and have a look. According to the sequence of base pairs in DNA strand, different genetic information are stored. So, sequence of base pairs. The features of organisms are determined by the sequence of base pairs. Accordingly, a gene is a specific base sequence in a DNA molecule responsible for a particular character. In other words, gene is a specific segment of DNA for a particular character. You have to know what a gene is. Then, the genes that determine large number of features in an organism and transmit them from generation to the next are present in chromosomes. Each gene has a particular location in a chromosome. So that is the concept of gene and the structure of the genetic material and the structure of DNA. So I am sure you all can understand that clearly. So with that, students, I am going to end this chapter, and in the next chapter we will start off the chapter by discussing what gene linkage is. That is different from the observations that Mendel obtained from his experiment. So we will try to understand the other concept, gene linkage.
To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.